Hello, and thank you for joining us for this presentation and discussion on the COVID-19 vaccine. My name is Du Bois Bowman, and I'm privileged to serve as Dean of the University of Michigan School of Public Health. The devastating impacts of the COVID-19 pandemic have been felt around the globe. As the world turns its attention to the landscape of COVID-19 vaccines, and we're all collectively wondering, is the end of this pandemic finally in sight, many open questions remain. I'm honored to introduce my colleague, the world-renowned epidemiologist, Dr. Arnold Monto, to help shed some light on what we know about the vaccines. Joining Dr. Monto today is Dr. Emily Martin, an associate professor of epidemiology at Michigan Public Health, and another colleague who's been working tirelessly to use her expertise to protect the public's health throughout this pandemic. In a year that has challenged all of us in countless ways, I've repeatedly said that the community of faculty, staff, and students at the University of Michigan School of Public Health gives me hope. Dr. Monto and Dr. Martin are no exception. With that, I'll turn it over to Dr. Emily Martin.
Good afternoon, and thank you for joining us today for this important and timely webinar. We are grateful for your participation today, and I'm sure you're all looking forward to hearing from our colleague, the world-renowned epidemiologist, Dr. Arnold Monto. So I'm going to keep my early comments here brief. Through the event today, you'll have the opportunity to ask questions in the Q&A function. The presentation will also be available for viewing on YouTube after the session. My name is Emily Martin, and I'm an associate professor of epidemiology at the University of Michigan School of Public Health. Today, I'm honored to introduce Dr. Monto, the Thomas Francis Collegiate Professor of Public Health at the University of Michigan School of Public Health. Dr. Monto is the chair of the FDA's Vaccines and Related Biological Products Advisory Committee that handles COVID-19 vaccine reviews. Throughout his distinguished career spanning 7 decades, Dr. Monto has been involved in pandemic planning and emergency response to influenza and other respiratory virus outbreaks, including the 1968 Hong Kong influenza pandemic, avian influenza, SARS, MERS, and of course, the coronavirus pandemic. Dr. Monto was recruited to Michigan Public Health in 1965, where he has worked closely with Thomas Francis Jr. on the landmark Tecumseh Community Health Study. Dr. Monto expanded the study's scope to look at the spread of respiratory infections in the 10,000 residents of Tecumseh, Michigan. And then during the 1968 influenza pandemic, Dr. Monto found that vaccinating school-aged children reduced infection in the entire community, an early demonstration of how herd immunity works. Since then, Dr. Monto has been involved in evaluating a variety of strategies to control influenza, including vaccines, antivirals, but also other interventions that have become all too familiar for us this year, like social distancing and face masks. In the 2000s, he was involved in developing pandemic control strategies, leading to work at the World Health Organization and in the US during the 2009 swine flu pandemic. He's led clinical trials establishing the superiority of inactivated vaccines compared to live attenuated vaccines for influenza. In 2010, Dr. Monto established the Household Influenza Vaccine Evaluation, or HIVE study, designed to allow the study of many aspects of respiratory virus infections over time. And this study has provided the foundation for a continued study of coronaviruses over time. In 2017, Dr. Monto was the plenary speaker of an NIH-led workshop to develop a strategic plan and research agenda aimed at the development of a universal influenza vaccine. This work has formed the foundation of the now Michigan Influenza Center, which launched as the pandemic was beginning. These are just a few highlights from Dr. Monto's impressive and groundbreaking career, and it is clear why he was selected to play a critical role in ensuring that we have access to a safe and effective COVID-19 vaccine as soon as possible. We at the University of Michigan School of Public Health are grateful for Dr. Monto's leadership and expertise in these challenging times. So without further ado, I will turn it over to Dr. Monto for his presentation.
Thank you so much, Emily. And I want to let everybody know that Emily is the co-director of the Michigan Influenza Center as we go forward. And we have decided not to change the name because we still want to remind people that influenza is a critical in what we are going to be looking at in the future. Now, Emily mentioned the hive study. And as we go into the winter, one of the things we saw when we did an analysis of the coronaviruses back in the about 8 months ago was that we found that they were very seasonal. Now, there were 4 seasonal coronaviruses, and they show up every winter. And we wondered whether the seasonality of these viruses was going to tell us something about the way SARS-CoV-2 or the COVID-19 was going to spread. And I think we're beginning to sense that what we are seeing now may in fact be a reflection of this kind of seasonal spread. Now, having introduced the need, because we know how badly we need vaccines and other pharmaceuticals to control this infection, let's start looking at what we're really supposed to be talking about today, and that is how we are going to be able to control this pandemic through vaccines. And we've all heard about Operation Warp Speed. And for those of us who don't have a background having watched Star Trek, this sounds kind of strange and suggests that there's undue speed associated with the development. And in fact, that's not the case. What is happening is that we are putting into a 1-year period what typically takes maybe 15 to 20 years in terms of vaccine development. And how are we doing that? Well, we're working off platforms that have been developed for other agents, often for the original SARS virus or for MERS or for other viruses to get things going. We are also telescoping the studies where we would have been doing things in sequence, and instead we're doing them in parallel. For example, we would never think of manufacturing and getting vaccines ready to go before they were tested and proven. The reason this is happening is because there's been a lot of attention to this, and also a lot of funds given to this effort. So this is one of the major success stories of the response to the pandemic. So what I want to convince you of today is that we are doing exactly what we ordinarily do with vaccine development, but on a shorter timeline. Now, this is an example of the kinds of things that are going on at the US level in terms of trying to get the vaccines out quickly. And it's basically a public-private partnership, and NIH in particular is taking advantage of a lot of the networks that have been established to test vaccines. And that's why we've been able to do these very large studies that are currently underway or just finishing on evaluating the efficacy of the vaccine. This gives you a very broad picture of what is going on. They're taking networks, for example, that were testing HIV vaccines and other vaccines and enlisting them to test the vaccines against COVID. This is a much more careful look, or look in detail at the strategies, and Operation Warp Speed is between departments of the federal government. It's the planning body for getting everything done, including distribution of vaccines to the states. The active is the one that we are going to be focusing more on today in terms of their activities, the results of these studies that are being put together mainly by NIH, and I'll go into this activity of active in greater detail. And then we have the networks that are doing the studies, and this is what's going on in active. And basically they are harmonizing the efficacy trials They're putting together the networks to do the studies. And most importantly, they have a unified data and safety monitoring board that is looking at all of the studies other than the Pfizer study. The Pfizer study has their own independent data safety and monitoring board. So whenever you hear something coming from there, from about a hold or something like that, about safety or breaking codes or anything like that, it's coming from a single data safety and monitoring board. Now, why are we able to move so quickly? And you've all read the press about how successful the vaccine has been in early trials or in trials where the code has been broken. And it's because immunity seems to be related to a specific protein, the spike protein, which allows entry of this virus into the cells. And we are indeed fortunate that this spike protein was identified earlier in work with SARS and MERS related coronavirus as the important component of the vaccine so that development could move quickly. And all of the vaccines that are being developed, whatever the platform, whatever the way they have been produced, are related to the spike protein. And that is the key to the strategy for vaccine control and prevention. Now, there are several different ways. I've illustrated 3 different ways of getting the body to produce antibodies and to respond in other ways with cellular immunity to this spike protein. The Moderna and Pfizer BioNTech strategy is to use mRNA. This is a genetic message. The AstraZeneca and later Janssen approach is to use a vector to attach the genetic material of the spike protein to an adenovirus vector. This is a virus which causes common colds and other illnesses in humans. And to not get antibody developed to that, they've used a chimpanzee adenovirus. And then we have another system where we use a more traditional approach, getting the protein of the spike into the body through a new method, which is to make, to get genetic material to make in cell culture the material that is going to result in production of antibodies and other responses. And if you see Sanofi at the bottom, this is very similar to what they are doing for a licensed flu vaccine called FluBlock. So these are different ways of getting the spike in so the body can respond to the spike protein. And this is just a quick summary of all the ways that vaccines can be produced. Why is RNA taking the lead right now? Because it is fast. It's much faster than other methods because what you're doing is putting the genetic material into the body and getting the body to respond. And we have non-replicating vectors, which is the adenovirus related vectors. And then we have the protein based, which is the third method I was talking about. And that is slower. And we are seeing this playing out in terms of the timetable for vaccine availability. Now, again, this is the nucleic acid vaccines, and there's the RNA at the bottom there, and the RNA is injected into the muscle, and it makes the body produce the viral protein to which the body responds. So it's all associated with producing SARS-CoV-2 spike and getting the body to respond. And here's another paper showing the chimpanzee adenovirus. If you look at the top, that's the code for the chimpanzee adenovirus. And there is another example of the method that is being used to get the protein in through a spike nanoparticle. Again, the spike keeps coming up. That's the critical part. Now, why is this going so quickly and efficiently? Well, we've talked about the laboratory development of the product that's going to be delivered. There have been standard assays of purity and lack of toxicity. This is standard for development of any vaccine, but it is being done quickly and efficiently because there's been a lot of resources given to it. Studies in small laboratory animals for spike antibody production, then in many cases, injection into non-human primates who were then challenged with the wild virus, and they've been protected. And that gave everybody great confidence in moving ahead to the large-scale studies that are being carried out. Phase 1 and phase 2, dose finding to find the better dose, because as I'll point out in just a minute, there's also been some issue of mild but clear side effects in terms of aching and in some cases fever associated with these vaccines. So you got to get the dose right. Now this is antibody, and I'll go through this very quickly because what's been remarkable about these vaccines is they all produce good antibody to the SARS-CoV-2 spike. And this is from the Pfizer vaccine, mRNA vaccine. And here is another example, and this is a little easier to see. And this is from the AstraZeneca adenovirus vector vaccine. And you can see what's going on here. And what you can also see is you don't get as good a response to a single dose as you do from 2 doses. So the 2-dose strategy has been just about universally used. We do think from some of the data in the Pfizer studies that you may get protection earlier before you get the 2nd dose, but you're not as fully protected until you get the 2nd dose. Side effects. There are mild, acceptable side effects to these vaccines. If anybody has got the zoster vaccine, the shingles vaccine, you know that those are reasonably reactogenic vaccines, and you know that you've gotten them because you may have a little aching and fever afterwards, especially after the 2nd dose. These are adverse reactions to the mRNA vaccines, and you can see the same kind of mild and moderate reactions to the adenoviral vaccines. And this is part of the dose finding. So you try to figure out what's tolerable in terms of side effects and what gives you the best vaccine response in terms of antibody. Safety assessments. We are very concerned about safety, of course, with anything that's new, that's anything that is using a novel platform, which hasn't been licensed before, which is the case with 2 of the 3 approaches we've talked about. And the only difference in safety between what we're doing now and what is traditionally done is if we go with emergency use, we're only going to have the time period from the dosage to the availability of the vaccine to go on. So we are going to have to keep watching. There's no reason to think that with this kind of efficacy that we're seeing, we're going to have any safety problems. But as you go to larger numbers, you have to have that in mind. Now, The collective DSMB, Data Safety Monitoring Board, did see something with the AstraZeneca vaccine, and they put a hold on. And that just shows how well this has been done in terms of trying to monitor the vaccine and how well it works. Now I'm going to use from now on some PowerPoints that were lent to me by the people at FDA to tell you about the licensure process at FDA, because I think that's a major concern. And I'll go through them rather rapidly to allow more time for discussion afterwards. And this PowerPoint just says why we have to do clinical trials to be sure that we are not deploying a weakly effective vaccine. We want to do the best. We want to have a standard approach used. Now, the efficacy endpoints that are being used, and this is from the guidance that FDA issued, what was being looked at is COVID-19 disease of any severity. They could have used preventing severe disease. They could have used preventing asymptomatic infection, but most have decided to use COVID disease of any severity. And what they established as the endpoint for approval would be greater than 50% efficacy, not realizing that the efficacy was going to be as high as has currently been seen. And among the secondary endpoints was going to be prevention of serious disease. And again, they thought that this would be questionable, and they put a lower bound of the confidence interval at 0%. And what we're going to see with the Pfizer vaccines is that there was near 100% prevention of serious disease. Clinical trial populations have to include racial and ethnic minorities. This has been done and will be part of the reports that everybody is going to see when we have our meeting next week. And the safety database needs to be large. They say 3,000 here. Well, we've got 30,000 or 40,000 within these trials. Again, money talks, and there have been a lot of investments put into vaccine development, which has resulted in large numbers of people being involved, which has had a lot of, so we have a lot of data. And not only do we have a lot of data, we also have a great deal of observations on clinical disease. And that's one of the reasons we've got to this point where we are able to look at emergency use very quickly. Now, emergency use, originally the statements were vague about when emergency use was going to be issued. But what we can say now is that they are using the same standards for efficacy for emergency use authorization. The only difference is that we are only having an average, a median of 2 months from the second dose. So there's a shorter duration. And what we're going to have to do is watch what happens going forward. There is no indication that we're going to have any surprises, but obviously you don't know what's going to happen as you go forward. And safety monitoring is going to continue, whether you have full licensure, emergency use authorization, and CDC and FDA are responsible for that. And we here with Emily are going to be part of CDC studies following the use of the vaccine in emergency use, and then later on after full licensure. So we're going to be doing all of these things and following up with both licensure and emergency use through observational studies. Now, I'll just very quickly talk about the Vaccines and Related Biological Committee, and I'm going to just skip a few of these PowerPoints just to allow more time for discussion. And this is the VRBPAC. And the FDA uses the VRBPAC in an advisory function. They generally agree with the recommendations of the VRBPAC. And we held a meeting on October 22nd to talk about the general principles of the vaccine approvals, to agree that FDA is setting these up right in terms of emergency use and full licensure. And there was discussion about this, and the issue was the 2 months for emergency use. And as I mentioned, the question of 2 months versus a longer period came up. It was decided that given the fact that we're in an emergency, that people are developing severe disease that needs to be prevented, that 2 months was a reasonable time to begin deployment of vaccines. And there were discussions of the primary endpoint. Should we be using more severe disease? Should we be using asymptomatic infection? And it was decided that the issue of asymptomatic infection could best be handled in post-licensure studies or post-availability studies, because vaccines are rarely assessed in terms of how they prevent asymptomatic infection. But we need, as we all know, to get a handle on the role of asymptomatic infection in transmission. Pediatrics, and this, it was a little bit of a more complicated discussion because we've seen some complications of infection, of SARS-CoV-2 infection in children. So the vaccines may need to be evaluated a little differently as we move into children. So we do not expect very quick approval for children based on the results in adults, bridging studies, so-called, but there may need to be further studies in children. Now, we all are concerned about distribution of vaccines, and that's not what the VRBPAC does. And yesterday, the Advisory Committee on Immunization Practice came up with recommendations for use of the vaccine. And this is a PowerPoint from the VRBPAC, from the ACIP at CDC, how they make their decisions. And this is again from the ACIP based on limited doses being available. And now we are told that these will be available to healthcare workers who have direct contact with patients, and also nursing home residents and people who care for nursing home residents. When larger doses are available, then the availability, the recommended target groups will be increased, and then we will have continued use. So when we talk about the end game, where we will be, it will probably be in late winter into the spring, when larger numbers of doses of vaccine are available and can be administered. And we can discuss that when we want to think about the end game. What is the end in sight? Now, just in closing, I want to let you know that our next meeting, the meeting of the VRBPAC, is on December 10th. This will be specifically to review the Pfizer vaccine for emergency use authorization. This is the web page that you can access, and as the previous meeting was on live on YouTube, this meeting will be live on YouTube. And unfortunately for me, sitting as chair, it's going to go from 9 a.m. to 6 p.m. I hope we can finish early, but what you're going to be able to find on December 8th is the report from Pfizer, the complete report from Pfizer attached to the web page. So you can see how the vaccine has done and how well it has prevented and the safety data. So nothing is being hidden. This is all an open, transparent process. And thank you all for listening. And I look forward to a robust discussion.
Thank you, Arnold. Wonderful presentation. So before we get into a discussion now, Arnold and I can debate vaccines and everything COVID and respiratory viruses from now until 2 o'clock, but we want to see everybody else's questions. So at the bottom of your screen, there is an option to choose the Q&A function, and there is where you can put questions in there. People will be watching, and I'll be watching for questions that we can add to prime our discussion. So Arnold, are you ready for?
I am ready.
All right. First question. So, there's been a lot of discussion, especially following the ACIP meeting yesterday, about prioritized groups for vaccination, who's going to get the vaccination and when. But there's a lot of people that aren't seeing themselves discussed in those conversations, right? Because we're talking about who are the first batches of vaccines are going to come on. But what would be your best guess for people that aren't in the priority groups, the first priority groups, in terms of when they could see a vaccination? So people like a healthy 65-year-old, a healthy 30-year-old, where's your guess in terms of when that's actually going to be available?
Well, I think we all are working from the same information availability which is basically in the media. Whereas there's a great deal of transparency about the FDA process, there's been less transparency because there are commercial considerations about the availability of doses. And one of the things that people don't realize is that this is a public-private partnership. And when FDA looks at this, they look at this as Pfizer making an application for a vaccine. For the same reason, there are issues about figuring out how many doses are going to be available and how many vaccines are going to be approved by that time. Because we know that Moderna is coming along. They're going to be discussed on December 17th. And then the next one's probably going to be AstraZeneca. But there have been some issues that have been in the media. It's hard to judge that. My best guess is that it's going to be into the late winter, early spring, before we're going to have enough doses available, because this is going to be driven by the number of doses available.
Along those lines, with in terms of the way the trials have gone, and the speed with which the fact that we're kind of compiling all of the questions for vaccine trial into one big trial for every product, what kind of safety, what does this mean for safety and efficacy data for populations where there would typically be a special trial just for this population? So one big example is pregnant people. We typically would have a trial just for pregnant people. What does this strategy mean for safety and efficacy data?
Well, I'm not sure that there's really typically a trial in pregnant women for most vaccines. In fact, most vaccines are used in pregnant women on the basis of observational studies. And part of that is because the companies typically do these trials and they're terribly concerned about liability as they do studies in pregnant women. The most recent studies that we've seen in pregnant women, and this is relatively unique, are RSV and influenza trials, as you well know, being directed to pregnant women, often to see if it protects their offspring against the particular illness. We are recommending vaccine for pregnant women for flu, a high priority at WHO, on the basis of mainly observational studies. And you and I do observational studies together. So we are convinced that we can learn a lot from observational studies. And I think as we go forward, with these COVID vaccines, given the fact that nobody will want to be in a placebo group anymore. We're going to have to rely on observational studies and good observational studies to give us the information we need.
Yeah, and I wonder how much the world is expecting how important observational studies are about to become.
I think they better, because we can't export our studies.
Yeah, yeah.
Once we've got, once we and this will apply to any improved vaccines. This is the first generation of COVID vaccines. We may have improved vaccines come up later on. And how do you study those?
Yeah.
You're going to have to study them in comparative trials and observationally.
Yeah.
And how are you going to be able to say which vaccine gives you longer protection? That's going to be, again, observational data.
It's going to look like the last couple of years of flu vaccine, where you have all these different products on the market and you're trying to figure out which one, everybody's trying to figure out which one should they drive to this pharmacy and get that vaccine, or that pharmacy and get that.
Right, and there's no incentive for companies to do this unless they're really convinced that they can make money and that their vaccine will prove superior.
Yeah. I'm gonna bring up a question that's a little bit on a little bit of a different topic now. As so there's been a lot of papers, particularly recently, on changes in the virus and genetic changes in the virus. And so a question for you is, what do you think, is that going to have any impact on vaccine effectiveness? Like we're so used to hearing with influenza, vaccine effectiveness being impacted by changes in the virus. So one, is that going to matter? And two, to what degree are we paying attention to other elements of the virus besides that one kind of spike site that we're mostly paying attention to?
Well, the first one's relatively easy, and as you well know, because we have our colleague Adam Loring, who's doing the sequencing with us. And while the virus is changing somewhat, it's not changing enough to escape from antibody produced to the virus that's messaged in the vaccine. Whether that's going to last, who knows? The positive is that we have been living with 4 seasonal coronaviruses, which cause common colds, and those viruses have been just about the same for the 50 years I've been involved in studying them. So we can still see the same genes associated with those viruses that produce antibodies. That's the good news. The bad news is that we do see reinfection with these viruses. And we need to figure out what the correlates of protection are, because people have a lot of antibody to the spike of these viruses. So first things first, we can stop the pandemic through use of the current vaccines and through, unfortunately, the herd immunity that's building up because of bad behavior. Bad behavior may be having good results in terms of getting natural infections, producing antibody in the population, which didn't happen last spring. And that, combined with a vaccine, is going to result, I would predict, in a decrease in transmission in our populations. And then we have to worry about improved vaccines, drift, all sorts of things. But we are indeed fortunate that the path to these vaccines was easy from a scientific standpoint, much easier than an HIV vaccine. Remember, they've been working on that for how many years?
It's been 10 years away for a very long time.
10 years, exactly. And also because of the resources that have been given to make people to de-risk the whole operation. They've de-risked companies and taken away any kind of liability, and that's in our society, that's what you need to get them to really move ahead quickly.
And it's, I've seen some questions come through about how can we assess duration of protection, especially given that. And that's going to be, I think that's sort of where the observational studies come in, that we're going to have to be watching carefully how long infection or how long protection continues after all of these vaccines are administered, even outside of clinical trials.
Right, and that will be relatively easy as long as we can get, as you and I both know, the data on who got what vaccines and things like that.
That's going to be the hardest part.
My concern is that a lot of effort, we've speeded vaccine development. We haven't put the same kind of resources into deployment and follow-up.
Yeah.
And we're going to need a lot of attention given to this to answer the critical questions that everybody wants to know, that you can't learn from the clinical trials, even if they involve 50 to 60,000 people. You're not going to learn everything in that kind of population when you're giving the vaccine to millions.
Yeah, and I think one of the big questions that's going to come up as this gets deployed is, now that we're starting to see reports about reinfection, which we know happens with the seasonal coronaviruses, and we've seen reinfection with the other coronaviruses. As we're starting to see cases of reinfection with this, how does that interplay with vaccination? And particularly, how does that help us answer people who are, so we usually tell people that have had COVID already, if you had it in the last 90 days, you don't need to get tested. You're probably protected for a little while. But should we be also vaccinating those people because of that potential for reinfection? What's your opinion on that?
My answer would be yes, but lower in the priority scheme. And the reason I say that is, paradoxically, the antibody being produced by these vaccines is better than what's been seen in people who have recovered from COVID. Whether that, and also, as you and I both know, that there are some weird things about asymptomatic infection, mild infection, not producing the kind of antibody titer that you see in severe infection, which is something we usually don't think of. So my answer to that is yes, they should be vaccinated, but since they certainly have partial protection, they're lower down on the priority list than others who have not been previously infected.
Yeah, this reminds me of when we did a seminar in January and we got asked about vaccination. We said a vaccination was going to have to beat nature, because a vaccination would have to be better than natural immunity to a coronavirus.
And we're not used to seeing that.
Not used to that.
This is, I keep coming back to calling this a weird virus. It's weird in so many ways. The disease it causes, the kind of transmission we saw in the spring, and even now the transmission when you're exposed. Some households will transmit to everybody, and some households there'll be practically no transmission, and we have super spreaders and non-spreaders. And with flu, we're much more, we can always predict it'll spread. With this, who knows?
Yeah, absolutely. Can we talk a bit more about side effects, and particularly side effects as they pertain to the fact that we've got all these different types of platforms? I mean, are the side effects coming out of the trials primarily like injection site, a couple of days, or are they long-term side effects that are being seen?
Well, they're not long-term side effects. I think, and interestingly, they seem to be associated more, at least with some of the vaccines, with the second shot than the first shot. I think the interesting thing will be to see if, when people give the baculovirus expressed vaccine, the protein vaccines, which are a lot more traditional in terms of not including genetic message and different platforms, adenovirus is getting in there. It's, even though it's not replicating, it's still doing things to T cells. What happens when we start going with those other vaccines? Is it a function, in other words, of the spike protein that's attached, or is it a function of the platform that's being used? We really don't know that yet.
Yeah, and I think, as we think about different platforms, there's a question about, and I've seen a couple of questions with this theme of when do you choose one versus, if you have the choice between multiple vaccines, which product should you choose? Are we going to know? And I mean, my viewpoint on this is that, we see this with influenza vaccines, you choose the one that's right there. And I think with the distribution and the production challenges, I wonder if what we're going to see is different products being deployed to different populations, different places. But me as an individual, I'm not going to say I have my choices between A, B, and C, at least in the early days.
Well, and then you have the minus 70 centigrade issue.
Yeah.
Which is going to limit distribution of the Pfizer vaccine. And I wonder what kind of future that vaccine is going to have as other vaccines emerge and are more easily administered.
Yeah.
It clearly will have an effect in much of the rest of the world. We look at some of our rural areas, but you have where most of the population of the world lives, there's no way a vaccine like the Pfizer vaccine can be administered there.
Yeah, absolutely. And then, it's nice to see that the Pfizer and the Moderna vaccines both have similar, their efficacy is around the same range, but if these other products come up with a different range of their efficacy, then we're going to have to figure out where we prioritize the more potent vaccines versus not. And it's kind of what we do with flu vaccine, right? We prioritize flu vaccines for certain populations, right?
And but the other thing is, let's hope enough people are given these other platform vaccines, because they may last longer. You may get a longer duration of immunity.
That's a good point.
It may be up and back. And that's why this is also important, because under emergency use authorization, it's a median of 2 months in terms of safety. It's also a median of 2 months in terms of efficacy.
Mm-hmm.
So they're very efficacious right after they're administered.
Yeah. Who
knows what happens long term?
It means you can't wait too long to get through a group of people too. You got to really get vaccine out there.
That's right.
Pockets of people.
But the need is there. And that's the rationale for the emergency use.
Yeah. All right. Changing topics again slightly. What are we going to do about the kids and the teens? Having an almost teen in the office next to me that would really like to go back to school.
Well, I'm not sure where this emergency use will end in terms of age. Some of the studies have been done down to 12 years of age. They have not been done in younger children. And as you well know, it's the teens that have been getting infected, and some of them have been getting sick. So I would predict that as we go forward, we may well slowly use the vaccines in older children, and then before we go to younger children, because it's the younger children mainly that we've seen this MIS-C, the multi-system inflammatory syndrome occurring with infection, and that's with infection. So who knows what happens if you give a vaccine, and there it may make a difference if you give a genetic material versus protein.
Yeah, I think there are some pediatric trials going on now, I think.
The pediatric trials really have taken have been put on the back burner in terms of prioritization. They're not, they don't have NIH pushing them the way that, and using all the networks.
Yeah, do you think that was because of the?
I think this is in part out of concern about moving too fast in children.
Do you think that maybe the early viewpoints that children weren't getting infected when it might have just been an epidemiologic phenomenon? But I think that may have also maybe set the stage for moving slower in children.
It did, but at the advisory, if there was any pushback about anything, it was about children's studies from the pediatricians in the group. They felt that the traditional approach of just using bridging studies, relatively small numbers of studies in children using production of antibody as the outcome, rather than looking at clinical protection of the rest, which you can't get in relatively small studies, should not be used. That larger studies should be carried out. And of course, as pediatricians, they said we should be doing, and I'm sure we will.
Yeah.
But the question is when.
When. So I think we have just a couple of minutes left. I'm gonna ask you one more question, and then let's see if there's anything else that we want to say before we close up. But so one of the things that I think, I think the American public is so used to flu vaccine campaigns, and flu vaccine campaigns are so centered around the one dose strategy, where you just find people wherever you can find them and you give them a dose and they're good for the season. Well, depending on how effective the flu vaccine is that year.
And then we hope it works.
We hope it works. This is a 2-dose strategy, which is operationally fundamentally different in how we think about things, especially when you have multiple products. And we want to get product A and then get product A again. So can you kind of explain for the audience why are we even going down this road? Why is there a 2-dose strategy? I know you touched on it in your presentation, but why are 2 doses important?
Well, 2 doses seem to be much more protective than 1 dose. Some of the studies may show, and they are looking at people who were infected during the clinical trials after the first dose, and we may see whether there is some level of protection. Right now, all of the data are for the 2-dose strategy. So it is important for people to get the second dose, and for healthcare providers to know what the first dose was so that they could give the right second dose, unless we have regional differences. So everybody in one region, because of availability, is getting one vaccine. In some other areas, they're getting another vaccine. But that's where we are now. We just have to remember, we didn't even have the sequence of this virus until January 2020, when the Chinese let us have it after not, and I won't go any further than that. But that was one of the first things we got around the 20th or so of January in 2020. Look how far we've gone since we got that sequence.
It's incredible. All right, so given that, when a vaccine is available for you, are you going to get it? And does that answer depend on which vaccine?
It won't depend on which vaccine. I will get it, but since I've been happily living, not going outside, I want to be sure that those people who are more at risk get it first. And then I will happily get the vaccine. I think what we've seen is much more encouraging. And you can tell how encouraging this is because of the 50% positivity that was set in the CDC, in the FDA guidance. And having this at greater than 90%, at least from the preliminary reports, really changes everything. And it should change people's attitudes. And it certainly should make, and they really haven't seen severe disease in anybody who has been vaccinated. Now, that may not continue because the most vulnerable, they've gone to older people, but they haven't gone to the most vulnerable. And some of these people may actually, because of comorbid conditions and the rest, have severe disease. But that sure is encouraging.
Yeah.
And I think that we should move ahead and get vaccinated when it's our turn to get vaccinated, because I think this is being looked at very carefully. And let's hope our our local authorities who are going to be under tremendous strain when they're handling the pandemic and now also trying to organize the distribution of vaccine, support them as best we can.
Well, definitely. I agree. My turn's going to come later than your turn, but I'll definitely get it when it's my turn. But I will, I think I might still use grocery delivery even after I get it, because I'm becoming used to not going to the grocery store and I enjoy it.
Well, my dear wife is in the other camp. She does not enjoy not being able to go into the grocery and pick out exactly what she wants. So we have one of our few diversions in opinion on that one.
Yeah. So when given all of this with the vaccine landscape and all of the distribution things, when do you this is a combination of both vaccine and this idea of vaccine driven herd immunity, when do you think life is going to feel normal again, or are we sort of forever changed as a society and how we interact with each other?
Well, I'm not going to go into the socioeconomic considerations, whether big office buildings will ever be filled the way they have been in the past, whether some of the things that we've done have changed. I'll just tell you my own view that it's so much more efficient when you and I are down the hall from each other and getting things done than having to communicate the way we're communicating now. And I think we'll be back to that level probably, if not in the middle of the spring, probably by early summer. Because I think we're going to see seasonality. I think it's going to be much more like like flu outbreaks, which behave themselves. This one has not behaved itself. I was surprised we had as much transmission over the summer as we did, and it's probably because we had nobody immune. So we're going to have a lot of people immune. We may not ever see stopping of transmission, at least in the middle of the winter, but things are going to be a lot more like normal by the time we get into warm weather.
I hope so. All right, well, with that, I am going to close today's session on that note. Thank you again, Arnold, for the presentation and the conversation. A link to this recording is going to be shared with all registrants, and it's also going to be posted on the School of Public Health website. It's going to be posted on YouTube for viewing and sharing later, and it's going to be available on the School of Public Health social media platforms. So please share widely and get this information out there. Thanks everybody for submitting your great questions and engaging, and thanks for coming to the talk today.